Have you ever wondered whether it's possible for early life stress to leave long-lasting molecular changes in the body? I'm Purya Vadapur, and as part of the Barham Lab at UCI, I try to answer this question in our own specific way. First and foremost, we need to start with a description of the body's primary stress response pathway. We know that there is a region inside the hypothalamus called the PVN, or the paraventricular nucleus, and inside the PVN, there are certain neurons that can be activated by stress. After they are activated, they release a hormone called CRH, or corticotropin releasing hormone, and through a series of cascades involving the anterior pituitary and the adrenal glands, CRH ultimately results in the secretion of cortisol, which is the body's primary and most commonly known stress hormone. On top of this, we also know that there are a class of molecules inside the body that are known as alarmins. And alarmins are essentially signaling molecules that are released as a response to stress. One specific example of an alarmin is HMGB1, which is a high mobility group box 1. And typically in regular cells or neurons, HMGB1 resides in the nucleus. As soon as a cell realizes that there is stress, whether it's physical or chemical stress, or other types of stress also, this HMGB1 is translocated out of the nucleus into the cytoplasm, and then it's actively secreted. Now, up until this point, there haven't been any research projects specifically looking for whether or not it's possible for early life stress to leave long-lasting changes in terms of the translocated HMGB1. And to do so, to answer this question in this research, we look at mouse models, specifically from the period when they were born, which we call postnatal day 0 or P0, up till P8. And we have a control group, and we have another group that has undergone early life stress in the form of limited bedding and nesting, which has been shown to result in psychological stress, which can also activate all the stress response pathways that I previously mentioned. So that's what we're looking at. And ultimately, we understand that there are no significant differences in terms of the translocated HMGB1, regardless of sex or exposure to early life stress. But we suggest that a future experiment with a larger sample size could better account basically for the variances in data that we noticed. Thank you for your attention.